Hello everybody, my name is Fernando from University College London, and I'm going to present our work on simulation of brain resections for quantitative analysis of epilepsy surgery. Epilepsy is a chronic disease of the brain that affects around 50 million people of all ages, which makes it one of the most common neurological diseases globally. It can often be treated with drugs. However, about 1/3 of epilepsy patients are drug resistant, and the way to cure these patients is by resecting the part of the brain that causes the seizures. This image shows a coronal slice of a postoperative T1 weighted MRI, where the right temporal lobe has been resected, as shown by the green arrow. However, only half of the patients who undergo a resective surgery are seizure-free after 5 years. Therefore, we need to better understand and predict who will be seizure-free after the surgery, and for this we need to know exactly how much of each brain structure has been removed. To quantify the resective surgery, the resection cavity must be first segmented from the post-op MRI, bottom left. Post-op MRI is registered to the pre-op MRI, and the segmented cavity is projected onto the pre-op space using the resulting transform. Finally, a brain parcellation obtained from the pre-op MRI is masked using the transformed cavity segmentation. The masked parcellation can then be used to compute the percentage of each resected brain structure. In this talk, I will focus on the method we developed to segment the cavity from the postoperative MRI. Cavity segmentation is a difficult problem due to the variability in shape and texture of the cavities and to the different postoperative phenomena that make some parts of the image unexpectedly dark and unrelated to the cavity. It is also not trivial to compare the pre- and postoperative images using registration due to the missing correspondences and nonlinear deformations. Here, green and red arrows show parts of the images that do and do not correspond to resection cavities, respectively. In the first case, on the left, white matter hypointensities make it difficult to differentiate white matter from CSF in the cavity. In the second case, the brain has shifted after the surgery, generating a new cavity filled with CSF. In the second and third cases, a postoperative edema has also appeared. In the third case, there is also a hyperintense blood clot within the cavity. The fourth case shows an arachnoid cyst adjacent to the resection cavity, which has a similar appearance to it. We could use deep learning to solve this problem. However, there are no publicly available annotated datasets, and manual segmentation is challenging, mostly because of the lack of intensity gradients between resection cavities and other areas filled with CSF, as shown by the yellow arrows in these two images on the right. To generate large dataset for training convolutional neural networks, we have developed a method to simulate resections on normal brain MR images. We generate an image of a resected brain and the corresponding ground truth segmentation from a normal T1 MRI and the corresponding brain parcellation that can be computed using standard software such as FreeSurfer. I will now explain how we generate the cavity shape, the ground truth label image, and the resected image. To generate the cavity shape, we first model a sphere as a geodesic polyhedron built from an icosahedron that can be subdivided using any frequency, depending on the number of vertices we'd like to obtain. The sphere is a mesh whose vertices will be radially displaced using a gradient noise to perturb the surface. If we use a uniform noise distribution, like the one on the left, changes in perturbation between neighboring vertices are too large and therefore not realistic. Using a smooth gradient noise, such as the one on the right, results are more realistic. Pearly noise is a type of gradient noise typically used to generate natural-looking signals such as handwritten lines, clouds, terrain in 3D games. We use simplex noise to perturb the sphere vertices, which is a variation of Pearly noise that is more computationally tractable for a large number of dimensions and generates fewer directional artifacts. Simplex noise is computed on each vertex of the sphere, and they are radially displaced accordingly. In this figure, noise values have been computed for all voxels for visualization purposes. Negative noise values are shown in blue, and positive values are shown in red. Finally, a random affine transformation is applied to the mesh for increased stochasticity. To generate the ground truth label segmentation, we start from a normal T1 weighted MRI and a corresponding brain parcellation. Gray matter binary mask from one of the hemispheres is computed from the parcellation, and a random voxel in it is selected as the center of the perturbed sphere. This is because the epileptogenic zone is always in the gray matter, therefore all resections must include this tissue. Then a resectable tissue mask is computed from the parcellation by excluding certain brain structures and applying different morphological operations. The ground truth label is computed as the intersection of the surface with the resectable tissue mask. Masking out the voxels in the MRI with the ground truth label generates an unrealistic simulation that could lead to a poor generalization to real cases. Therefore, we use a CSF mask computed from the parcellation to generate a CSF-like image that is blended with the original MRI using a randomly smoothed version of the ground truth label as alpha channel in order to mimic partial volume effects near the boundaries. As the simulation takes only a couple of seconds, we run it on the fly during training, which allows for large variability in the training set. The simulation is implemented as a torch-IO transform, and the code is available at these repositories. To test the efficacy of a method, we have used data from two different sources. First, we collected images from 430 subjects that underwent resective surgery at our institution and performed manual segmentation of the resection cavity on 133 of them, plus 67 from 2 other human raters to assess inter-rater variability. We also downloaded more than 1,800 T1 weighted MR images from the OASIS, ICSI, and ATNI datasets for training. We simulate resections during training on the images in the green rectangle. The yellow rectangle corresponds to the EPISURG dataset. I'll now explain some of our experiments. First, we train a baseline model with our 133 manually segmented images using tenfold cross-validation. Then we use the publicly available images to train using only simulated data for self-supervised learning. We also added the preoperative images in EPISURG to test whether using images from the test distribution was beneficial. Finally, we used the model from point 3 to create pseudo-labels for the unlabeled postoperative images from EPISURG and train a new model from scratch using semi-supervised learning. We use the same custom implementation of a small 3D unit for all the experiments and test all models on the manually segmented images. We found that the baseline model performed quite poorly with a median die score of 65. Training using a larger simulated dataset, we obtain a median die score of 80, which is a statistically significant improvement with respect to the baseline. Adding the pseudo-labeled images resulted in a marginal improvement, obtaining a die score of 82. We can see that the score for all models and the inter-rater agreement are within the same range, except for the baseline. And here are some qualitative results. In these images, the manual annotations are shown in green and predictions of the best model are shown in magenta. We can see for the median case there are some discrepancies around the border, where it is difficult to define the resection boundaries. In the worst performing case, the model partially misinterpreted a region of brain shift as a resection cavity. And most failures are caused by brain shift or by blood clots within the cavities. To conclude, we have developed a method to simulate brain resection cavities on preoperative MRI images, and we train a convolutional neural network with simulated data, showing that results are superior if the dataset is large enough. The code is open source and available on GitHub. In the future, we will explore different simulation models to understand the necessary complexity to model the resection cavity. We will also explore additional semi-supervised learning techniques to better exploit our unlabeled postoperative images to improve results. I'd like to thank my colleagues and my funders, and thank you for watching.